Dear students, after studying this module, you shall be able to understand the usefulness of UV visible spectroscopy for quantitative and qualitative analysis. Initially, quantitative analysis using UV visible spectroscopy. The determination of concentration of a sample under study or analyte is one of the major applications of UV visible spectroscopy. Quantitative analysis involves the determination of exact concentration of analyte under study. The reason behind application of UV visible spectroscopy for quantitative analysis is that most of the organic and inorganic compounds strongly absorb the radiations in UV visible region of the electromagnetic spectrum. Even if the compound does not absorb the UV visible radiation, or if its absorbance is too weak, it can be converted into other compound by chemical reaction that absorbs strongly these radiations. For example, a dilute solution of Fe2+ ions does not absorb visible light. Reacting the ferrous ions with orthophenanthroline, however, forms an orange-red complex of ferrous phenanthrin 2+ and strongly absorbs the radiation near 500 nanometers. From this complex, concentration of Fe2+ ions can be measured. The quantitative analysis using UV visible spectroscopy is based on the Lambert-Beer's law of absorption, which relates the absorption with the concentration of analyte, where absorbance is equal to ELC. From the Lambert-Beer's law, by studying the absorbance concentration of analyte can be determined. The extinction coefficient E in this expression is characteristic of a given substance under study. However, value of extinction coefficient E depends upon the measurement conditions such as wavelength, solvent, temperature, and also on the characteristics of the instrument used. Because of these reasons, value of the extinction coefficient E is not used for the quantitative analysis. Quantitative analysis is often done by using calibration curve for the substance to be analyzed, which is constructed by using one or more standard solutions with the known concentration of the analyte. UV visible spectroscopy provides a highly accurate method for quantitative analysis. Let us try to understand why. UV visible spectroscopy involves the electronic transitions in which energy difference between the ground and the excited state is fairly large. Because of this, almost all the molecules are present in the ground electronic state at room temperature. It increases both the sensitivity and the accuracy of measurements as compared to the other methods of quantitative analysis, and thus provides the basis for thousands of quantitative analysis methods. To get the more accurate results, the sample to be analyzed must contain only absorbing component for which the calibration has been performed. Pure sample of the solvent must be used as a blank during the measurements. Quantitative analysis using chemical derivatization. There are many compounds that exhibit very weak or no absorbance in UV visible region. For these compounds, quantitative analysis using UV visible spectroscopy can be performed by chemical derivatization, that is, by formation of their derivative. This method involves the formation of a derivative of a given compound under study that exhibit very weak absorbance by using some organic reagent which forms a complex with strong absorptivity. The quantitative analysis of this complex can be performed in the same way as explained earlier using Lambert-Beer's law. In this method, proper choice of organic reagent can increase both the sensitivity and the selectivity of the quantitative analysis. The determination of iron in blood serum is one of the examples of this method. Human blood usually contains 45% by volume cells and 55% by volume plasma. If blood is collected without an anticoagulant, the blood coagulates and the liquid that remains is called serum. Serum normally contains iron attached to transferrin. This serum does not absorb the UV visible radiations. For spectrophotometric determination of iron in blood serum, firstly, ferric ions in the transferrin are reduced to ferrous ions, which is released from the protein by using a reducing agent such as hydroxylamine, ascorbic acid or thioglycolic acid. Then trichloroacetic acid is added to precipitate the proteins, leaving ferrous ions in the solution. A measured volume of supernatant liquid, which is a colorless solution containing ferrous ions, is then transferred to the fresh tube. Buffer and excess of ferrozine, which is a chromophoric reagent, is then added wherein a purple complex is formed. The absorbance is then measured at 562 nanometers, which is the lambda max for the given complex, from which the amount of iron can be determined as shown here in the graphical representation. Spectrophotometric titrations. Volumetric analysis is one of the important methods of quantitative analysis that involves the study of the color changes at the endpoint of a titration. This color change is often detected through visual examinations. This process is highly subjective and can be the source of error in analysis. The endpoint detection by the use of UV visible spectroscopy introduces the objectivity into the analysis and lends itself a automation. In photometric titrations, a spectrophotometer is used to detect the endpoints graphically. The absorbance of a titrated species is measured after each addition of a titrant until the endpoint is well passed. A plot of absorbance versus milliliters of the titrant is done, and it consists of the two straight lines that intersect at an endpoint of the titration. This method is called as mole ratio method. For a successful photometric titration, it is necessary that the measured species adhere to Lambert-Beer's law. For example, spectrophotometric titration of copper-2 with EDTA. The reaction for this experiment can be seen here, wherein a complex is formed, that is a sodium salt of EDTA. The titration is performed at 625 nanometers. Both the copper EDTA chelate and the copper 1 ion absorbs at this wavelength, but the molar absorptivity of the chelate is much higher. The typical graph are shown here in the figure 2 for the titration of copper 2 with EDTA and shows the marginal difference between the two. Enzyme kinetic assay. In case of biological systems, enzyme assays can be used in the indirect analysis of one compound or a group of compounds. By carefully selecting an enzyme, any change in the sample following the addition of enzymes will result from the reaction of the specific compound or compounds. Enzyme assays are based on the selectivity. Enzyme assays can be divided broadly into two types: rate assays and the endpoint assay. Rate assay: factors which affect the rate of an enzyme are temperature, pH, enzyme activity, enzyme concentration and the substrate concentration. If all the other parameters are controlled at the constant level, the rate of reaction is directly proportional to the substrate concentration. Endpoint assay. Here the conditions are selected in such a manner that the conversion of substrate to the product is completed within a reasonable time, that is around 5 to 20 minutes. The most common method of detection in enzyme assay is spectrophotometric assay. This assay uses a spectrophotometer, which is a machine used to measure an amount of absorbance of light by the substance. It then combines the kinetic measurements and the Beer's law by calculating the appearance of product or disappearance of the substrate concentrations. The difference between the initial absorbance and the final absorbance is directly proportional to the amount of substrate. This method is simple, non-destructive, selective and sensitive. For example, consider the NADH-NAD+ molecule, which is often used in the enzymatic oxidation reduction reactions. During these reactions, NADH is often oxidized to NAD+ or NAD+ is reduced to NADH. NADH absorbs light at 340 nanometers. However, NAD+ does not hold that property. A spectrophotometer can measure the change in absorbance of 340 nanometer light, thus indicating a change in amount of NADH. Quantitative analysis of drugs. Let us consider the case of drugs from the forensic point of view. Many drugs are either in the form of raw materials or mixtures, including in the form of formulations. Drugs can be assayed by making their solutions in a suitable solvent and measurements of the absorbance of the specific wavelength. For example, diazepam tablet can be analyzed by 0.5% sulfuric acid in methanol at the wavelength of 284 nanometers. Quantitative analysis of proteins in milk using UV visible spectroscopy. The quantitative analysis of proteins can be carried with UV visible spectroscopy by measurement of wavelength of absorbance in protein assay and using Beer-Lambert law. This law states that when a solute absorbs light of a particular wavelength, the absorbance is directly proportional to the concentration of the substance in solution. This method of protein assay is based on the development of color with the dye. Coomassie Blue G-250 binds to protein. It is also known as Bradford method. The unique chemical property of the dye makes it interact with the side chains or the R groups of the specific amino acids. There is a correlation between the blue color developed and the concentration of the protein in the sample. The more protein, the more intense the blue color. Quantitatively, the result is obtained using the UV visible spectrometer. Coomassie G-250 dye can bind to proteins and it absorbs more strongly at 595 nanometers. Hence, this particular wavelength is selected for Bradford assay. The light passes through the sample cuvette and some light is absorbed by the cuvette and its contents. The light transmitted is read by a detector, and by the comparison with the light transmission through the reference cuvette and the absorbance is then estimated. Colorimetric assays use calibration curves based on the absorbance of the standard solutions to determine the concentration of the unknown samples. Qualitative analysis using UV visible spectroscopy. UV visible spectroscopy provides a limited amount of qualitative information as spectra generally consist of only few broad absorbance bands as compared with the other techniques such as infrared spectroscopy or NMR spectroscopy, which produces many narrow bands. Most of the absorption bands in the UV visible spectra of the various results from the presence of the unsaturation in their structure. A chromophore is a molecular group which is responsible for absorption and usually containing an unsaturated bond. When inserted into a saturated hydrocarbon which exhibits no UV visible absorbance spectrum, it produces a compound with the absorption between 185 and 1000 nanometers. Some common chromophores and the absorption maxima for them are listed here in the table. For example, we can look into a carbonyl group. Example, acetone absorbs at the lambda max of 271 nanometers. A carbonyl group of an aldehyde, in the case of an acetaldehyde, absorbs at 293 nanometers, and furthermore for the other chromophoric groups. Thus, from the presence of the absorbance band at a particular wavelength, it indicates the presence of a particular chromophore in that molecule. However, a position of absorbance maxima is not fixed even for a particular chromophore, as it depends upon the nature of the whole molecules, presence of the oxochromes, and also on the solvent in which the sample may be dissolved for recording a UV visible spectra. Experimental parameters such as temperature and pH value of solution also may affect the absorption maxima as well as intensity of absorption. Even though UV visible spectra do not provide the absolute identification of unknown compound, however, it can be used for the identity of substance through comparison of the measured spectrum with the reference spectrum if available. More qualitative information from the UV visible absorption spectra. UV visible spectroscopy can also used to determine the physiochemical properties of a compound, which provide useful qualitative information about that compound. This can be done by the following ways. Color. Color is an important property of a molecule and can be used for the identification of a molecule. The human eye sees the complementary color to which is absorbed, which has many limitations. UV visible spectrophotometers can be used to measure the color of a molecule and hence can be used for the qualitative analysis. Qualitative analysis of proteins. A protein largely shows an absorbance in the UV range due to the presence of aromatic amino acids such as tryptophan, tyrosine, phenylalanine, and shows a strong absorbance at 260 nanometers. A protein at a room temperature has a specific tertiary structure or conformations that in turn creates a specific electronic environment for the aromatic amino acids. If the protein is heated, it will at a certain temperature unfold or melt and lose its structure. This is called denaturation. In this process, the electronic environment of the aromatic amino acid changes, which in turn results in the spectral changes or shifts. Thus, UV spectroscopy can be used for the qualitative analysis of proteins and to study the denaturation of proteins. Detection of impurities. UV absorption spectroscopy is one of the best methods for determination of impurities in organic molecules. Additional peaks can be observed due to impurities in the sample, and it can be compared with that of a standard raw material. By also measuring the absorbance at a specific wavelength, the impurities can be detected. Benzene appears as a common impurity in cyclohexane. Its presence can be easily detected by its absorption at 255 nanometer. Some more applications of UV visible spectroscopy. Some other applications of UV visible spectroscopy are discussed. Detection of conjugation. With the help of ultraviolet spectrum, one can find whether a given compound contains a certain group in conjugation with each other or not. For example, 4-methyl pent 3-ene 2-one and 4-methyl pent 4-ene 2-one. Since 4-methyl pent 3-ene 2-one has the carbon-carbon double bond and the carbonyl group in conjugation with each other, so in this case the absorption will take place at a longer wavelength as compared to 4-methyl pent 4-ene 2-one, presented by structure 2, which does not have any kind of a conjugation. Thus, UV visible spectrum enables us to determine whether the compound contains conjugated system or not. Determination of configuration of geometrical isomers. In case of the geometrical isomers, trans isomer shows the absorption maxima at a longer wavelength and also have a larger molar absorptivity values as compared to the cis isomer. For example, trans-stilbene, as shown by figure 1, showed a lambda max at 294 nanometers with the molar absorptivity at 24,000, while cis-stilbene showed lambda max at 278 nanometers with the molar absorptivity of 9350. This observation can be explained on the basis of the steric hindrance. In trans isomer, two bulky phenyl groups are opposite to each other and there is no steric hindrance, as a result of which the double bond and the pi bonds of the phenyl rings lie in the same plane, and thus the pi orbital overlapping of the conjugated system is highly effective, as a result of which a trans isomer absorbs at a longer wavelength. Reversely, in the cis isomer, two bulky phenyl groups are present on the same side of the double bond, due to which there is a large steric hindrance. Because of this steric hindrance, double bond and the pi bonds of the phenyl ring are forced to be non-polar. Due to this, pi orbital overlapping is less effective, which results in decrease in conjugation, and hence the cis isomer absorbs at a longer wavelength. Determination of strength of hydrogen bonding. The strength of the hydrogen bonding formed by a compound with polar protic solvents such as water or alcohol can be studied with the help of UV visible spectroscopy by comparing the lambda max in protic solvent and in the non-protic solvent. For example, the carbonyl compound forms a hydrogen bond between water and carbonyl oxygen atom, as a result of which energy of the non-bonding electrons N of the carbonyl oxygen in the ground state decreases by an amount equal to the strength of the hydrogen bond. Therefore, N to pi star transition of the carbonyl group shifts towards the shorter wavelength. For example, N to pi star transition of the acetone in the non-polar solvent hexane takes place at 279 nanometers, but in the polar protic solvent water, it occurs at 264 nanometers. Thus, from this, hypsochromic shift of 15 nanometers is the strength of the hydrogen bond which can be calculated. Study of the keto-enol tautomerism. UV visible spectra can be used to study the keto-enol tautomerism, as both keto and the enol form of the substance shows a characteristic absorption bands that can be identified. For example, ethyl acetoacetate, which shows a keto-enol tautomerism, absorption band for the keto form has lambda max at 275 nanometers with the low absorption intensity, that is 16, while the enol form has a lambda max at 244 nanometers with the high absorption intensity of 16,000. To summarize, UV visible spectroscopy provides a useful method for quantitative analysis. Quantitative analysis using UV visible spectroscopy is based on Lambert-Beer's law. Quantitative analysis using UV visible spectroscopy can also be done by using chemical derivatization, spectrophotometric titrations, or by the enzymatic assay. UV visible spectroscopy, however, is less useful method for qualitative analysis as compared to the other methods. The less usefulness of this is due to the presence of only few bands in UV visible spectrum, and the position depends upon the environment of the whole molecule as well as the experimental conditions such as solvent, pH and temperature. UV visible spectroscopy also found an application in detection of conjugative systems, studying isomers like cis-trans and keto-enol etc., and to study the strength of the hydrogen bond.